afternoon, students. Today we have second lecture in physiology of digestion, and today we are going to talk about such questions: digestive effects of pancreatic juice, anatomic histological features of the hepatic biliary system, the composition of bile and its role in the digestion, control of bile production and its secretion in the duodenum, indigestive liver functions of the large intestine and control of its secretory activity, the role of the different digestive tract sections in the absorption and mechanism of absorption of different substances, and system mechanism of hunger and satiety, the concept of absorptive and post-absorptive phases of digestion. So let's start with anatomic functional features of the pancreas. So the pancreas is located retroperitoneal, posterior to the stomach, in the inferior part of the left upper quadrant. It consists of head located within the curvature of the duodenum, a body and a tail which extends to the spleen. The pancreas is a complex organ composed of endocrine and exocrine tissues performing several functions. The endocrine part of the pancreas consists of pancreatic islets, which are also called as islets of Langerhans. The islet cell produce the hormones insulin and glucagon, which enter the blood and which we were talking in our previous semester. And these hormones are very important in controlling blood levels of nutrients such as glucose and amino acids. The exocrine part of the pancreas is a compound acinar gland, and this is what we will talk today. So the acini produce digestive enzymes. Clusters of acini are connected by small ducts, which join to form larger ducts, and the larger ducts join to form the pancreatic duct. The pancreatic duct joins the common bile duct at the hepatopancreatic ampulla and empties into the duodenum at the major duodenal papilla. An accessory pancreatic duct present in most people opens at the minor duodenal papilla. A smooth muscle sphincter surrounds the pancreatic duct where it enters the hepatopancreatic ampulla, and there is a smooth muscle sphincter at the minor duodenal papilla. Pancreatic juice, the exocrine secretion of the pancreas, moves through the pancreatic ducts to the small intestine where it functions in digestion. Pancreatic juice has an aqueous and an enzymatic component. The aqueous component is produced principally by columnar epithelial cells that line the smaller ducts of the pancreas. Bicarbonate ions are a major part of the aqueous component, and they neutralize the acidic kind that enters the small intestine from the stomach. The increased pH caused by pancreatic secretions in the duodenum stops pepsin digestion, but provides the proper environment for the function of pancreatic enzymes. The enzymatic component consists of enzymes produced by the acinar cells of the pancreas. These enzymes are important for the digestion of all major classes of food. Without the enzymes produced by pancreas, carbohydrates, lipids, and proteins are not adequately digested. The proteolytic pancreatic enzymes digest proteins. The major proteolytic enzymes are trypsin, chymotrypsin, and carboxypeptidase. They are secreted in inactive forms, whereas many of the other enzymes are secreted in active form. They are secreted in the inactive forms as trypsinogen, chymotrypsinogen, and procarboxypeptidase, and are activated by the removal of certain peptides from the larger precursor proteins. If these enzymes were produced in the active forms, they would digest the tissues producing them. Pancreatic juice also contains pancreatic amylase, which continues the polysaccharide digestion initiated in the oral cavity. Pancreatic lipases, which break down lipids, and nucleases, which reduce DNA and ribonucleic acid to the component nucleotides. Pancreatic exocrine secretion is regulated primarily by hormonal mechanisms. A small amount of parasympathetically induced pancreatic secretion occurs during the cephalic phase of digestion, with a further token increase occurring during the gastric phase in response to gastrin. However, the predominant stimulation of pancreatic secretion occurs during the intestinal phase of digestion when chyme is in the small intestine. The release of the two major enterogastrons, secretin and cholecystokinin, in response to chyme in the duodenum, plays a central role in controlling pancreatic secretion. Of the factors that stimulate enterogastron release, which are fat, acid, hypertonicity, and distension, the primary stimulus, specifically for secretin release, is acid in duodenum. Secretin, in turn, is carried by the blood to the pancreas, where it stimulates the duct cells to markedly increase the secretion of aqueous fluid into the duodenum. Even though other stimuli may cause the release of secretin, it is appropriate that the most potent stimulus is acid, because secretin promotes the alkaline pancreatic secretion that neutralizes the acid. This mechanism provides a control system for maintaining neutrality of the chyme in the intestine. The amount of secretin released is proportional to the amount of acid that enters the duodenum. So the amount of sodium HCO3 secreted parallels the duodenal acidity. Role of cholecystokinin in pancreatic secretion. So cholecystokinin is important in regulating pancreatic digestive enzyme secretion. The main stimulus for release of cholecystokinin from the duodenal mucosa is the presence of fat and, to a lesser extent, protein products. The circulatory system transports cholecystokinin to the pancreas, where it stimulates the pancreatic acinar cells to increase digestive enzyme secretion. Among these enzymes are lipase and the proteolytic enzymes, which appropriately further digest the fat and protein that initiated the response and also help digest carbohydrate. In contrast to fat and protein, carbohydrate does not have any direct influence on pancreatic digestive enzyme secretion. All three types of pancreatic digestive enzymes are packaged together in the zymogen granules, so all the pancreatic enzymes are released together on exocytosis of the granules. Therefore, even though the total amount of enzymes released varies depending on the type of meal consumed, the most being secreted in response to fat, as I previously said, the proportion of enzymes released does not vary on a meal-to-meal basis. That is, a high protein meal does not cause the release of a greater proportion of proteolytic enzymes. Evidence suggests, however, that long-term adjustments in the proportion of the types of enzymes produced may occur as an adaptive response to a prolonged change in diet. For example, with a long-term switch to a high-protein diet, a greater proportion of proteolytic enzymes are produced. Cholecystokinin may play a role in pancreatic digestive enzyme adaptation to changes in diet. Just as gastrin is trophic to the stomach and small intestine, cholecystokinin and secretin exerts trophic effects on the exocrine pancreas to maintain its integrity. We will now look at the contribution of the remaining accessory digestive unit, the liver and gallbladder. So besides pancreatic juice, the other secretory product emptied into the duodenal lumen is bile. The biliary system includes the liver, gallbladder, and associated ducts. The anatomic organization of the liver permits each hepatocyte to be in direct contact with blood from two sources: arterial blood coming from the aorta and venous blood coming directly from the digestive tract. Like other cells, the hepatocytes receive fresh arterial blood via the hepatic artery, which supplies the oxygen and delivers bloodborne metabolites for hepatic processing. Venous blood also enters the liver by the hepatic portal system, a unique and complex vascular connection between the digestive tract and the liver. The veins draining the digestive tract do not directly join the inferior vena cava, the large vein that returns blood to the heart. Instead, the veins from the stomach and intestine enter the hepatic portal vein, which carries the products absorbed from the digestive tract directly to the liver for processing, storage, or detoxification before they gain access to the general circulation. Within the liver, the portal vein again breaks up into a capillary network to permit exchange between the blood and the hepatocytes before draining into the hepatic vein, which joins the inferior vena cava. The liver is organized into functional units known as lobules, which are hexagonal arrangements of tissue surrounding a central vein. At each of the 6 outer corners of the lobule are 3 vessels: a branch of the hepatic artery, a branch of the hepatic portal vein, and a bile duct. Blood from the branches of both the hepatic artery and portal vein flows from the periphery of the lobule into the large expanded capillary spaces called sinusoids, which run between rows of the liver cells, so the central vein like spokes on a bicycle wheel. The Kupffer cells line the sinusoids and engulf and destroy all the red blood cells and bacteria that pass through in the blood. The hepatocytes are arranged between the sinusoids in plates 2 cell layers thick, so that each lateral edge faces a sinusoidal pool of blood. The central veins of all the liver lobules converge to form the hepatic vein, which carries the blood away from the liver. The thin bile-carrying channel, a bile canaliculus, runs between the cells within each hepatic plate. Hepatocytes continuously secrete bile into these thin channels, which carry the bile to a bile duct at the periphery of the lobule. The bile ducts from the various lobules converge to eventually form the common bile duct, which transports the bile from the liver to the duodenum. Each hepatocyte is in contact with the sinusoid on one side and a bile canaliculus on the other side. On the other side. The bile is subsequently stored and concentrated in the gallbladder between meals. After a meal, bile enters the duodenum as a result of the combined effects of gallbladder, emptying and increased bile secretion by the liver. The amount of bile secreted per day ranges from 250 milliliters to 1 liter, depending on the degree of stimulation. Bile contains several organic constituents, namely bile salts, cholesterol, lecithin, and bilirubin, in an aqueous alkaline fluid, similar to the pancreatic sodium HCO3 secretion. Even though bile does not contain any digestive enzymes, it is important for the digestion and absorption of fats, primarily through the activity of bile salts. Bile salts are derivatives of cholesterol. They are actively secreted into the bile and eventually enter the duodenum along with the other biliary constituents. Following the participation in fat digestion and absorption, most bile salts are reabsorbed into the blood by special active transport mechanisms located in the terminal ileum. From here, bile salts are returned by the hepatic portal system to the liver, which resecretes them into the bile. Bile salts aid fat digestion through the detergent action, emulsification, and facilitate fat absorption by participating in the formation of micelles. Both functions are related to the structure of bile salts. The term detergent action refers to bile salts' ability to convert large fat globules into a lipid emulsion consisting of many small fat droplets suspended in the aqueous chyme, thus increasing the surface area available for attack by pancreatic lipase. Fat globules, no matter their size, are made up primarily of undigested triglyceride molecules. To digest fat, lipase must come into direct contact with the triglyceride molecule. Because triglycerides are not soluble in water, they tend to aggregate into large droplets in the watery environment of the small intestine lumen. If bile salts did not emulsify these large droplets, lipase could act on the triglyceride molecules only at the surface of the large droplets, and fat digestion would be greatly prolonged. A bile salt molecule contains a lipid-soluble part, a steroid derived from cholesterol, plus a negatively charged water-soluble part. Bile salts absorbed on the surface of a fat droplet, that is, the lipid-soluble part of the bile salt dissolves in the fat droplet, leaving the charged water-soluble part projecting from the surface of the droplet. Intestinal mixing movements break up large fat droplets into smaller ones. These small droplets would quickly recoalesce were it not for bile salts absorbing on the surface of each droplet and creating a shell of water-soluble negative charge. Because like charges repel, these negatively charged groups on the droplet surfaces cause the fat droplets to repel one another. This electrical repulsion prevents the small droplets from recoalences into large fat droplets, and thus produces a lipid emulsion that increases the surface area available for lipase action. Now let's talk about the micelle formation. So bile salts, along with cholesterol and lecithin, which are also constituents of bile, play an important role in facilitating fat absorption through micelle formation. Like bile salts, lecithin has both a lipid-soluble and water-soluble part, whereas cholesterol is almost insoluble in water. In a micelle, the bile salts and lecithin aggregate in small clusters with the fat-soluble parts huddled together in the middle to form a hydrophobic core, while the water-soluble parts form an outer hydrophilic shell. Micelles are water-soluble because of the hydrophilic shells, but they can dissolve water-insoluble substances in the lipid-soluble cores. Micelles, those provide a handy vehicle for carrying water-soluble substances through the watery luminal contents. The most important lipid-soluble substances carried within micelles are the products of fat digestions, as well as fat-soluble vitamins, which are all transported to the sites of absorption by this means. If they did not hitch a ride in the water-soluble micelles, these nutrients would float on the surface of the aqueous chyme, just as oil floats on the top of water, never reaching the absorptive surfaces of the small intestine. In addition, cholesterol, a highly water-insoluble substance, dissolves in the micelles hydrophobic core. This mechanism is important in cholesterol homeostasis. Bile secretion by the liver may be increased by chemical, hormonal, and neural mechanisms. So let's talk about chemical mechanisms by the help of bile salts. Any substance that increases bile secretion is called a choleretic. The most potent choleretic is bile salts themselves. Between meals, bile is stored in the gallbladder, but during a meal, bile is emptied into the duodenum as the gallbladder contracts. After bile salts participate in fat digestion and absorption, they are reabsorbed and returned by the enterohepatic circulation to the liver, where they act as potent choleretics to stimulate further bile secretion. Therefore, during a meal, when bile salts are needed and being used, bile secretion by the liver is enhanced. So what is the hormonal mechanism? It occurs by the help of secretin. Besides increasing the aqueous sodium HCO3 secretion by the pancreas, secretin stimulates an aqueous alkaline bile secretion by the liver ducts without any corresponding increase in bile salts. Neutral mechanisms by the help of vagus nerve. Vagal stimulation of the liver plays a minor role in bile secretion during the cephalic phase of digestion, promoting an increase in liver bile flow before food ever reaches the stomach or intestine. During digestion of a meal, when chyme reaches the small intestine, the presence of food, especially fat products, in the duodenal lumen triggers the release of cholecystokinin. This hormone stimulates relaxation of the sphincter of Oddi and contraction of the gallbladder, so bile is discharged into duodenum, where it appropriately aids in the digestion and absorption of the fat that initiated the release of cholecystokinin. Now let's talk about undigested functions of the liver. So these are participation in the metabolism of carbohydrates. So the liver plays an extremely important role in maintaining normal blood glucose levels. When this level is lowering, the liver uses glycogen stores to replenish blood glucose. This process is called glycogenolysis. In addition, substances such as amino acids, lactic acids, fructose, and galactose may be converted into glucose in the liver if necessary. In the case of excessive glucose concentration in the blood, the liver converts it into glycogen and triglycerides. Next one function, participation in the metabolism of fats. Hepatocytes can accumulate a small amount of triglycerides and use them as an energy source for ATP synthesis, if necessary. In the liver needed for the transport of fatty acids, triglycerides and cholesterol to and from cells lipoproteins are synthesized. The liver is capable of synthesizing cholesterol to convert it into bile acids. Participation in metabolism of proteins. Hepatocytes take part in the deamination of amino acids, creating opportunities for the oxidation or conversion to glucose or fatty acids. The toxic product of deamination, ammonia, is converted by the liver into a less toxic substance, urea, which is excreted by the kidneys in the urine. The majority of plasma proteins are synthesized in the liver, in particular albumins, globulins, prothrombin, and fibrinogen. Removal of bilirubin from the body as a component of bile. Bilirubin is a product of the conversion of heme, a component of hemoglobin, protein which is excreted from destroyed old red blood cells and is absorbed by the liver from the blood. In the intestine, most of the extracted from the bile bilirubin is metabolized by bacteria, and the rest of it is excreted with the feces, giving them a special yellow color. Next function, chemical transformation of hormones and drugs taken. The liver detoxifies exogenous substances such as ethyl alcohol, antibiotics, sulfonylamides, and others. Hepatocytes metabolize and excrete endogenous hormones with bile in the intestine, in particular iodinated thyroid hormones, estrogens, and aldosterone. Next one, participation in the exchange of vitamins and trace elements. Hepatocytes are deposit and secrete into the blood, if necessary, a number of vitamins. So the liver is the primary depot for such vitamins as A, B12, D, E, K, as well as some minerals, in particular iron and copper. Along with the kidneys and skin, the liver takes part in the conversion of vitamin D into its active forms. And the last function is phagocytic Cooper cells of the liver. They are capable of phagocytosis, of destroyed all erythrocytes, leukocytes, and even bacteria in the case of blood contamination. Anatomical and functional features of the large intestine. So the large intestine consists of the colon, cecum, appendix, and rectum. The cecum forms a blind-ended pouch below the junction of the small and large intestines at the ileocecal valve. The small finger-like projection at the bottom of the cecum is the appendix, which is a lymphoid tissue that houses lymphocytes. The colon which makes up most of the large intestine, is not coiled like the small intestine, but consists of three relatively straight parts: the ascending colon, the transverse colon, and the descending colon. The end part of the descending colon becomes S-shaped, forming the sigmoid colon, and then straightens out to form the rectum. What are the functions of the large intestine? So first is secretion of intestinal juice with high mucus content, which facilitates the adhesion of indigested residues and the formation of feces, and has a protective effect in relation of the mucous membrane. Reabsorption of water from the chyme, which helps to thicken the feces and periodically remove them from the intestine defecation. It is inhabited by a microorganism which participate in the hydrolysis of some components of the chyme, which are not subjected to digestive processes in the small intestine, in particular cellulose and oxidation of undigested carbohydrates and root and undigested proteins. Synthesis some amino acids like histamine and tyramine and vitamins of B and K groups, create an immunological barrier in relation to pathogenic microorganisms that enter the intestine in the chyme content, participate in the metabolism of phospholipids, bile and fatty acids, cholesterol and bilirubin, provides the act of defecation. Now let's talk about the regulation of motility of large intestine. Most of the time, movements of the large intestine are slow and non-propulsive, as is appropriate for its absorptive and storage functions. The colon's main motility is haustral contractions initiated by the autonomous rhythmicity of colonic smooth muscle cells. These contractions, which throw the large intestine into pouches or sacs called haustra, are similar to small intestine segmentations, but occur less frequently. 30 minutes may elapse between haustral contractions, whereas segmentation contractions in the small intestine occur at rates of between 9 and 12 per minute. The location of the haustral sacs gradually changes as a relaxed segment that has formed a sac slowly contracts while a previously contracted area simultaneously relaxes to form a new sac. These movements are non-propulsive. They slowly shuffle the contents in a back and forth mixing movements that exposes the colonic contents to the absorptive mucosa. Haustral contractions are largely controlled by locally mediated reflexes involving the intrinsic plexuses. 3 to 4 times a day, generally after meals, a marked increase in motility takes place, during which large segments of the ascending and transverse colon contract simultaneously, driving the feces 1/3 to 3/4 of the length of the colon in a few seconds. These massive contractions, appropriately called mass movements, drive the colonic contents into the distal part of the large intestine, where material is stored until defecation occurs. When food enters the stomach, mass movements are triggered in the colon primarily by the gastrocolic reflex, which is mediated from the stomach to the colon by gastrin and by the extrinsic autonomic nerves. In many people, this reflex is most evident after the first meal of the day and is often followed by the urge to defecate. Thus, when a new meal enters the digestive tract, reflexes are initiated to move the existing contents further along the tract to make way for the incoming food. The gastroileal reflex moves the remaining small intestine contents into the large intestine. And the gastrocolic reflex pushes the colonic contents into the rectum, triggering the defecation reflex. Some absorption takes place within the colon, but not to the same extent as in the small intestine. Because the luminal surface of the colon is fairly smooth, it has considerably less absorptive surface area than the small intestine. Furthermore, the colon is not equipped with extensive specialized transport mechanisms as the small intestine is. When excessive small intestine motility delivers the contents to the colon before absorption of nutrients has been completed, the colon cannot absorb most of these materials and they are lost in diarrhea. The colon normally absorbs salts and water. Sodium is actively absorbed. Chloride follows passively down the electrical gradient, and water follows osmotically. The colon absorbs token amounts of other electrolytes as well as vitamin K synthesized by colonic bacteria. Through absorption of salts and water, a firm fecal mass is formed. Of the 500 milliliters of material enter the colon per day from the small intestine, the colon normally absorbs about 350 milliliters, leaving 150 grams of feces to be eliminated from the body each day. This fecal material normally consists of 100 grams of water and 50 grams of solid, including undigested cellulose, bilirubin, bacteria, and small amounts of salts. Those contrary to popular thinking that the digestive tract is not a major excretory passageway for eliminating waste from the body. The main waste product excreted in the feces is bilirubin. The other fecal constituents are unabsorbed food residues and bacteria, which were never actually a part of the body. Bacteria account for nearly 1/3 the dry weight of feces. Now let's talk about mechanism of absorption. So carbohydrate absorption, dietary carbohydrates are presented to the small intestine for absorption mainly in the forms of disaccharides maltose, sucrose, and lactose. The disaccharadases located in the brush border membrane of the small intestine cells further reduce these disaccharides into the absorbable monosaccharide units of glucose, mostly galactose and fructose. Glucose and galactose are both absorbed by the secondary active transport, in which symport carriers such as the sodium and glucose co-transporter on the luminal membrane transport both the monosaccharide and sodium from the lumen into the interior of the intestinal cell. Glucose or galactose, having been concentrated in the cell by these symporters, leads the cell down its concentration gradient by facilitated diffusion to enter the capillary network within the villus. Fructose is absorbed into the blood solely by facilitated diffusion. Mechanism of the lipids absorption. When the stomach contents are emptied into the duodenum, The ingested fat is aggregated into large oily triglyceride droplets that float in the chyme. Recall that through the bile salts detergent action in the small intestine lumen, the large droplets are dispersed into a lipid emulsification of small droplets, exposing a greater surface area of fat for digestion by pancreatic lipase. The products of lipase digestion, monoglycerides and free fatty acids, are also not very water-soluble, so little of these end products of fat digestion can diffuse through the aqueous chyme to reach the absorptive lining. However, biliary components facilitate absorption of these fatty end products by forming micelles. Fat absorption, remember that micelles are water-soluble particles that can carry the end products of fat digestion within the lipid-soluble interiors. Once these micelles reach the luminal membranes of the epithelial cells, the monoglycerides and free fatty acids passively diffuse from the micelles through the lipid component of the epithelial cell membranes to enter the interior of these cells. As these fat products leave the micelles and are absorbed across the epithelial cell membranes, the micelles can pick up more monoglycerides and free fatty acids, which have been produced from digestion of other triglyceride molecules in the fat emulsion. Bile salts continuously repeat the fat solubilizing function down the length of the small intestine until all fat is absorbed. Then the bile salts themselves are reabsorbed in the terminal ileum by special active transport. This is an efficient process because relatively small amounts of bile salts can facilitate digestion and absorption of large amounts of fat, which with each bile salt performing its ferrying function repeatedly before it is reabsorbed. Once within the interior of the epithelial cells, the monoglycerides and free fatty acids are resynthesized into triglycerides. These triglycerides conglomerate into droplets and are coated with a layer of lipoprotein synthesized by the endoplasmatic reticulum of the epithelial cell, which makes the fat droplets water soluble. The large coated fat droplets, known as fatty acids, from the chyme across the luminal membrane of the small intestinal epithelial cells, is a passive process because the lipid-soluble fatty end products merely dissolve in and pass through the lipid part of the membrane. However, the overall sequence of events needed for fat absorption requires energy. For example, bile salts are actively secreted by the liver. The resynthesis of triglycerides and formation of chylomicrons within the epithelial cells are active processes, and the exocytosis of chylomicrons requires energy. Chylomicrons are extruded by exocytosis from the epithelial cells into the interstitial fluid within the villus. Chylomicrons are 75 to 500 nanometers in diameter, compared to micelles, which are 3 to 10 nanomicrometers in diameter. The chylomicrons subsequently enter the central lacteals rather than capillaries because of the structural differences between these two vessels. Capillaries have a basement membrane and outer layer of polysaccharides that prevents the chylomicrons from entering but the lymph vessels do not have this barrier. Those fat can be absorbed into lymphatics, but not directly into the blood. The actual absorption of the monoglycerides and free fatty acids from the chyme across the luminal membrane of the small intestinal epithelial cells is a passive process because the lipid-soluble fatty end products merely dissolve in and pass through the lipid part of the membrane. However, the overall sequence of events needed for fat absorption requires energy. For example, bile salts are actively secreted by the liver. The resynthesis of triglycerides and formation of chylomicron within the epithelial cells are active processes, and the exocytosis of chylomicrons requires energy. In the stomach, the enzyme pepsin breaks down proteins, producing shorter amino acid chains called polypeptides. Only about 10–20% of the total ingested protein is digested by pepsin in the stomach. The remaining proteins and polypeptide chains leave the stomach and enter the small intestine. The pancreatic enzymes trypsin, chymotrypsin, and carboxypeptidase continue the digestive process and produce small peptide chains. Peptidases, which are enzymes bound to the microvilli of the small intestine, break down the small peptide chains into triglycerides, 3 amino acids, dipeptides, 2 amino acids, and single amino acids. At least 7 different transport systems are responsible for the movement of amino acids into intestinal epithelial cells. The predominant transport system moves most neutral amino acids by secondary active transport. A sodium-potassium pump establishes a concentration gradient for sodium from the outside to the inside of epithelial intestinal cells. A importer then moves amino acids along with sodium into the cell. Other transport systems move acidic and basic amino acids. The sodium concentration gradient is used by antiporters to establish a hydrogen concentration gradient. A importer then moves 3 peptides and dipeptides along with hydrogen ions into the cell. The total amount of each amino acid that enters the intestinal epithelial cells as 3 peptides or dipeptides is considerably more than the amount that enters as single amino acids. Once inside the cells, 3 peptidases and dipeptidases split the 3 peptidase and dipeptides into the component amino acids. Individual amino acids then leave the epithelial cells by facilitated diffusion or other methods and enter the hepatic portal system, which transport them to the liver. The amino acids may be modified in the liver or released into the blood and distributed throughout the body. And the last topic, which we will be discussing today, is endocrine control of hunger and satiety feelings. So the arcuate nucleus of the hypothalamus plays a central role in both the long-term control of energy balance and body weight, and the short-term control of food intake from the meal to meal. The arcuate nucleus is an arc-shaped collection of neurons located adjacent to the floor of the third ventricle. Multiple, highly integrated redundant pathways crisscross into and out of the arcuate nucleus, indicative of the complex systems involved in feeling of satiety. Feeding or appetite signals give rise to the sensation of hunger, driving us to eat. By contrast, satiety is the feeling of being full. Satiety signals tell us when we have had enough and suppress the desire to eat. The arcuate nucleus has two subsets of neurons that function in an opposing manner. One subset releases neuropeptide Y, and the other releases melanocortins derived from pro-opiomelanocortin, a precursor molecule that can be cleaved in different ways to produce several hormone products. Neuropeptide Y, one of the most potent appetite stimulators ever found, leads to increased food intake, thus promoting weight gain. Melanocortins, a group of hormones traditionally known to be important in varying the skin color for the purpose of camouflage in some species, have been shown to exert an unexpected role in energy homeostasis in humans. Melanocortins, most notably melanocyte-stimulating hormone from the hypothalamus, suppress appetite, thus leading to reduced food intake and weight loss. But neuropeptide Y and melanocortins are not the final effectors in appetite control. These arcuate nucleus chemical messengers, in turn, influence the release of neuropeptides in other parts of the brain that exerts more direct control over food intake. Scientists are trying to unravel the other factors that act upstream and downstream from neuropeptide Y and melanocortins to regulate appetite. Based on current evidence, the following regulatory inputs to the arcuate nucleus and beyond are important in the long-term maintenance of energy balance and the short-term control of food intake at meals. Other hormones that control the hunger and satiety feelings are leptin. So this hormone is secreted by adipocytes in proportion to the mass of accumulated adipose tissue. It is able to penetrate the blood-brain barrier, stimulate anorexigenic neurons, and suppress orexigenic neurons, thus reducing appetite. Insulin. It has a similar effect to leptin, but its effect is short-term and manifests itself periodically with an increase of insulin secretion in response to an increase in blood glucose. Next hormone, glucagon-like peptide 1. This hormone is secreted by L cells of the intestine when chyme is rich on triglycerides and carbohydrates and affects the arcuate nucleus, similar to leptin and insulin, suppressing appetite. The YY peptide is also secreted by L cells of the intestine when mucosal is irritated with chyme containing a big amount of nutrients. This hormone has a direct effect on the hypothalamic neurons, reducing the sense of hunger and inhibits the secretion of the hormone ghrelin in the stomach. Ghrelin, this is the most powerful humoral stimulator of hunger. It is secreted by the endocrine cells of the gastric glands in the empty stomach and stimulates the orexigenic neurons and suppresses anorexigenic neurons, increasing appetite and eating. Especially a lot of it is secreted during fasting and weight loss. Thank you for your attention. See you next week.